Good evening, everyone. It's a tremendous pleasure to introduce Pam Soltis. Pam is a distinguished professor and curator of the Florida Museum of Natural History at the University of Florida. And Pam is, by all accounts, a brilliant scholar and leader in the field of plant evolutionary biology. So, Pam, it's a real treat that you could be here tonight.
Thank you.
Thank you. So Pam currently leads the renowned Laboratory of Molecular Systematics and Evolutionary Genetics at the University of Florida, where scientists collaborate on a variety of questions investigating mechanisms of plant speciation and evolutionary relationships, especially among flowering plants. And I think it's actually worth taking sort of a step back, though, and really recognizing and acknowledging the sheer breadth, both in terms of the organisms of interest and the questions in which her lab is focused. So they really span all of land plants. So here we're talking about huge groups of species across deep evolutionary times, but they also focus on very recent patterns of speciation. It's quite dynamic. Pam received her BA in biology, summa cum laude at Central College in Iowa, and she went on to complete both of her advanced degrees, master's and PhD at the University of Kansas, which is a real hub of organismal research. And again, her research includes investigations of the evolution of flowering plants. She's also interested in the conservation of genetics of rare plants, and as well as historical processes that govern the geographic distribution of plants. She's really considered a pioneer in her work understanding the origin and evolutionary significance of chromosome evolution in plants, and she's been at the forefront of really aggregating as well as producing enormous amounts of especially molecular data and synthesizing them in novel ways to understand plant evolution. She's received a ton of awards, too many really to go through, but I did highlight 4 here that were worth mentioning. The first is effectively the sort of botany equivalent of the Nobel Prize, and that is the 2002 Dahlgren International Prize in Botany from the Royal Physiographic Society of Lund, Sweden. She's also received, in 2006, the Asa Gray Award from the American Society of Plant Taxonomists, the Botanical Merit Award, the Botanical Society of America of Plants Merit Award in 2010, and in 2004, Thomson Reuters gave her the title of World's Most Influential Scientific Mind. Astonishing, really. Not surprisingly, she's published hundreds, I didn't actually run the citation index, but of articles, peer-reviewed in top-tier journals like the American Journal of Botany, Systematic Biology, Science, Proceedings of the National Academy of Sciences, and Nature, as well as co-authoring several books, most recently a textbook on the ordering of plant biodiversity. She's also co-edited books on the developmental genetics of the flower, polyploidy and genome evolution, and molecular systematics of plants. 1 and 2, I think. I actually remember as a student not far over there reading those texts. But she's also garnered an astonishing number of awards, primarily from the National Science Foundation, as well as the Mellon Foundation, totaling millions of dollars. I was actually I benefited from one of those collaborative projects. It was actually my only source of funding as a postdoctoral fellow when I was at the University of Michigan. But I think importantly, in addition to being just an amazing scholar all around, she's also an incredible mentor and has demonstrated a real ongoing commitment to teaching undergraduates, graduate training, and I think especially the promotion of young women in science. So the list of accomplishments go on. I won't continue, and instead I'll please welcome Professor Pam Soltis.
Well, thanks a lot, Chuck, for that wonderful introduction. I guess I will just say that I have a lot of collaborators, and so anything that seems to be sort of a large contribution is really because of having a really active lab and a lot of people who work with me. So I'd like to make sure that that's all clear. So, well, good evening, everyone. Thanks so much for coming out on, yes, a chilly evening, although I understand it's not quite so chilly to you as it is to me, perhaps. But I'm getting used to it. I grew up in the Midwest, so I can handle this, I think. Okay. Oops. All right, so I'd like to start out by just talking a little bit about biodiversity. And biodiversity is the totality of life on Earth. Its physical forms, its genetic forms, its functional forms. And in all, we currently have about 1.8 million named species. This includes about 500,000 plant species. However, we understand that this is just the tip of the iceberg. There may be 10 times as many species, or maybe 100 times as many species as we've currently named. And most of our information about all of these species actually comes not directly from nature, but from our natural history collections. And in the US alone, we have probably over 1,600 natural history collections. And these house potentially 1 to 2 billion specimens. Worldwide, it's estimated that there may be 3 to 4 billion specimens housed in our natural history collections. And these collections can tell us incredible things about the past history of life on Earth as we attempt to incorporate this information into various analyses of molecular data, chemical data, and so on. Now, for centuries, our natural history collections have primarily been applied to studies of systematics and taxonomy. That is, these specimens allowed us to compare species and make inferences about how species might be related to each other. And in fact, it's interesting that most new species descriptions come not from new collections, but from new analyses of specimens that are housed in natural history collections. There was recently an analysis of some insect collections across Europe in which 4 new species were described simply from an analysis of the specimens in those collections. But we now recognize that these natural history collections can provide us with information on a host of other sorts of topics, ranging from genetics, even to genomics, to chemistry, to species interactions, to ecology, phenology, biogeography, and so on. And so these natural history collections, such as those housed in this building, can provide us with amazing new perspectives on evolution and the history of life. In addition, these specimens can help us as we address the biodiversity crisis and various societal problems. So for example, understanding patterns of loss of biodiversity can best be accomplished when we have the historical records that these natural history collections can provide. Just as one example, invasive species have tremendous ecological and economic impacts, and it's estimated that they cost the US over $120 billion per year. They're particularly devastating in areas such as Florida, where I live, because of our extensive coastlines and lots of shipping and our combination of temperate and subtropical climates. However, we have a very difficult time getting a handle on the factors that are involved in the spread of invasive species. And if we don't understand how they spread, it's very difficult to curb the invasions. And so we think that through the application of historical records from herbaria and other natural history collections, we can start to get a better handle on how these species might spread in their introduced habitats. Now, unfortunately, though, most of our natural history collections have specimens that are essentially locked away from most scientists and certainly from the public. However, recent analyses and recent initiatives have promoted the use of natural history collections through digitization. So digitization, that is putting our natural history collections online, is actually leading to a lot of new opportunities to use our natural history collections. Most of the digitization has involved label data. That is, if this is our herbarium specimens, such as some of the ones located over there to your left, most of the specimens will have a label, and on this label we'll have the scientific name, the date, the collector, the location where that specimen was collected, associated species, various notes, and so on. And this information has been transcribed in many cases and is being made available online. Now, in addition to the label data, however, though we're also interested in capturing not just the data, but the images of the specimens as well. And so a number of different sorts of mechanisms have been developed for imaging natural history collections. And just a few of these are shown here. So we have our herbarium specimen here on the right. Herbarium specimens are, in fact, one of the easiest things to digitize because they're essentially flat pressed plant specimens. So a simple photograph often will suffice. But there are various other types of digitization, image capture, that are also taking place, and a few of these other representatives are shown here. So for example, some 3D reconstructions here. We have some paleo collections for many insects. It's not individual specimens that are imaged, but in fact whole drawers of insects may be photographed and then served online. And so we have a number of different ways in which people are starting to capture the information found in our natural history collections and put them online. Now, thanks to a new program at the National Science Foundation, more and more US collections are actually going online. Our university is now home to IDIG Bio, which is the National Coordinating Center for the Digitization of Biodiversity Collections. And this project is allowing us to ingest from various collections the locality information, images, and other data, and then serve the data to scientists and the public. And then associated with this is the opportunity to develop new ways in which these data can be used for scientific research and for education. And in all cases, we're interested in the locality data, the dates, the images, and in some cases, based on the digitization project, even the vocalizations, for example, of birds and of amphibians. Now, the motivating factor behind this new initiative that NSF has sponsored is basically research. What new research questions can be addressed by digitizing and making this information available? So currently, there is a set of 13 projects designed around a specific research theme, and these encompass over 200 institutions that range across the country. And so currently, there are data coming into IDIG Bio from these 200 institutions in all 50 states. The topics of some of these research networks are listed here. I've highlighted the first one here, Flowering Time and Climate Change in New England Plants, and this is certainly one of the projects that Chuck's group is involved with here. The Harvard Herbaria are involved with digitizing information and then recording the dates at which species have flowered, and then this is being tracked across centuries, in fact, to identify changes in flowering time associated with climate change. And then, of course, we have a few other examples listed here as well, where bryophytes and lichens, for example, are being used as indicators of climate change across North America. There's a project monitoring the spread of invasive species in the Great Lakes region, and then another project that's involved with pairing up specimens of plants, their insect herbivores, and the parasitoids that parasitize those herbivores. And so this is a multi-trophic layer analysis, which is leading to some really exciting new developments in how we put specimen data together to enable new sorts of research. And again, there are 13 of these projects that are now underway that are designed to harness the power of these natural history collections. Now, for the most part, these projects are based on the label data that I described before, but more and more of these projects will be incorporating aspects of the specimen itself, maybe some aspects of morphology, that is the general appearance and shape of the specimens. Now, all of this information is being stored in a cloud computing environment, and the scale of this project has led to the development of new information technology procedures and technologies as well. And so our project is actually a collaboration between biologists and computer scientists in order to enable this huge influx and serving of biodiversity data. Now, in case you might be interested in finding out more about IDIG Bio, our homepage is shown here along with our URL, idigbio.org. And what you'll see here is that we, in fact, have over 26 million specimens now assembled within this data structure. This is a 10-year project. We're into year 4 at this point, and the community has really come together to digitize so much information, and we're now making that available. Now, it's possible from the IDIG Bio homepage to go to our specimen portal, and the specimen portal is the place where one can query our database to ask if there's information on a species of interest. So you can see again here, we have over 26 million specimen records, over 4 million media records, most of which are images, but more and more increasingly are also vocalizations and other sorts of data, films as well as the static images as well. So let's say we wanted to actually search for specimens for a given species. We could go to our search page, and let's say we're interested in searching for red maples. So we could enter in under the scientific name here, Acer rubrum. At this point, we don't have applications that will allow for common names, but we're working on developing a link to some common names so that people who are interested in looking up something that they might be familiar with from a local natural area might be able to look those up by the common name as well. But currently, it's based on scientific names only. And so we get the return. This will tell us how many specimens there are in the database, and I can't read what that says, but there are hundreds. And we also get a map here, which when we blow it up, will show us the distribution of those specimens. Some of the quality of the specimens varies. So for example, we probably really do not have a red maple growing out here somewhere in the Atlantic east of Jacksonville. But in fact, the majority of these specimens are probably more or less accurately described in terms of their distributions. What you may also notice, if you know red maples, you'll know that they're an eastern species. There may also be some red maples that have been planted in other parts of the continent, or these may actually represent the herbaria where these are housed. So a word of warning, you don't want to take any of these sorts of distributional data at face value. There would always be an element of data cleaning where you actually evaluate the quality of the data. But what this allows us to do very easily is access information from herbaria that are located across the country. And until very recently, this sort of enterprise was not possible. So if we select a given specimen record, we can find out more information about it. Each specimen record has the information about that particular specimen. It also tells us where that specimen is housed, which herbarium it's located in, or which museum collection if it's not a plant specimen. And it also tells us whether or not there are any associated media, for example, photographs. And so you can then click on that and get an enlarged version, and then also get more information about the specimen itself. So this is essentially our capability at the moment. We're working to enhance the mapping attributes of the portal, and we're also in the process of linking this information with other sorts of data. So far, essentially, a specimen is in the data set more or less in isolation. So that specimen may be linked in some way to its image, its a vocalization if there is one, perhaps a DNA sample, which is something that we're working on. But what we're really interested in doing is integrating the specimen information with various other sorts of databases that are available on a national or international scale. And so we'd like to be able to link these specimens, our natural history collections, to other sorts of resources. For example, resources on phylogeny or evolutionary history, and I'll talk more about that in a few minutes. Also, linking them to ecology, to paleontology, to our living collections, particularly to genomics and our huge databases that we have in terms of gene sequences, and then various other repositories. And so we think actually that these linkages, which we're just starting to explore at this point, between IDIG Bio and any of these other sorts of repositories that are listed here, will allow us to investigate some new and exciting research questions that to this point we've not really been able to explore. So the question is then, what can we do with specimen data? In the past, as I mentioned, specimens have been largely confined to studies of systematics and taxonomy. But now, as we start to think more about how locality information, the dates of collection, and so on may contribute to new sorts of evolutionary studies, we can see that there are really a lot of opportunities for addressing a wide array of questions. So we can monitor shifts in biodiversity through time, we can track invasive species, and so on. And the one that I want to spend a few minutes talking about is what's referred to as ecological niche modeling, particularly in association with climate change. And here I'll present a specific example from our own lab as a way to illustrate this. So we're interested in evaluating what might happen to the vast diversity of plant life in Florida under models of climate change. There are currently over 4,000 species of plants that occur in Florida. We have this great combination of warm, temperate, and subtropical climates that come together to create unique opportunities and unique habitats. And this has led to some amazing diversifications. So what we're interested in is taking information from the herbaria in Florida and elsewhere, and using that information to help us understand what might happen to that diversity as the climate changes over the next several decades. And this is work that is being conducted primarily by Charlotte Germain Aubry, who's a postdoc working with me, and also with huge contributions from Julie Allen, who's a postdoc at the University of Illinois. Now, in this project, we're modeling the distribution of these plant species at the current point in time, and then we want to project those models of where those species might occur into the future. So we're taking location information from herbarium specimens and coupling that with environmental data that we can extract from various databases, things like temperature, precipitation, soil types, and so on, are particularly important aspects of environmental data. And then we can use some specific software to model the range of each species. Now, there are a number of sets of software that are available for doing this sort of work, but we've actually found that for certain reasons, many of these approaches are really inadequate for what we're interested in doing. And so we've actually had to develop some new methodologies along the way. And this, of course, has, in a way, slowed us down, but also in a way helped us to devise much better models, we think, for where these species are best suited under current conditions and where they might occur in the future. So we're developing these models, and then we project them onto the future climate conditions as predicted by climate modelers. So we've currently developed models for about 2,500 species, but I'm only going to report on some models for about 1,600 of these. We've used over 500,000 data points that have come from herbarium records, by the time we do all of that evaluating of whether or not the records are good records or bad records, we've cut that almost in half. Our actual data going into the models is more like 300 and some thousand specimens rather than the full set. And then again, we're using environmental features such as temperature, precipitation, and soil to help develop models. So what does this look like? Well, we see as we model species and project them into the future that there will be some ecological winners and some ecological losers. The first example here is Albigardia ovata, the flat spike sedge, which is a relative of the grass family. And you can see a picture of it here. It's currently distributed primarily in southern Florida, and this heat map shows that this is the optimal habitat in this area. You'll also note, I'm sure, that this is also a highly urbanized area. So despite the fact that that represents the ideal habitat, the habitat is probably very limited in terms of what's really available to this species. Now, as we project a hotter and drier Florida for the year 2050, you'll see that that heat map is much cooler. And in fact, the amount of optimal habitat is going to be quite limited as we move forward. Now, in contrast, we have the scrub plum here on the bottom panel. This is a relative of the cultivated plum. And currently, it's distributed in this area called the Lake Wales Ridge. And we'll come back to that area a little bit later, but I do want to point out that we do actually have a little bit of topography in Florida. This is a ridge. It gets up to about 300 feet, and when you're on top of it, you can actually tell that you're on a ridge. And this is an amazing area because it was actually the only part of Florida that was not underwater during various parts of recent glacial cycles. And so there's a unique flora and fauna that is found along that ridge. Now, it's hot, it's dry, and it has very specific sandy soils that are found primarily there, but not only there, across the state of Florida. Now, you can see, though, as we go into a hotter and drier Florida, that in fact, the scrub plum might do very, very well. This area in central Florida with the optimal habitat is going to expand, as will other parts of the state as well. So it's difficult to predict whether a species will do well or do poorly under models of climate change. So we've made these models and projections for, as I mentioned, about 1,600 species. And this is a summary of the plant diversity now, where the green areas represent high species diversity and the tan areas represent low species diversity. Now, as we model the future and we look to a hotter, drier 2050, I won't show you the results for 2080, but they're even more extreme, we see that there are species movements that are anticipated. In this map, the green areas represent an area where there's an influx of species, and the tan areas represent those where species will be departing from. So we see that there will be species moving into the northern part of the state as expected for temperate regions in the northern hemisphere. As climate gets hotter and drier, species are projected to move northward. But we also see some increased movement southward into sort of the Everglades region here, where it is projected to be at least wetter than where it might be in other parts of the state. So we see perhaps an unanticipated bidirectional projected movement of plant species, one set of species moving northward and another set of species moving southward. Now, of course, this doesn't take into account the fact that sea levels will also be rising and affecting the habitats of Florida. There are some models that predict that by 2050, there could be as much as a 4-meter increase in sea level. And although most models are more in the 1 meter, this is a map showing what would happen with a 4-meter increase. And in this map, all of the gray areas will actually be underwater. So recall that this is one of those areas where the plant species are projected to move into, sort of from the central region of the state. And in fact, there will not be habitat available at that particular point in time. So this paints a not very optimistic view of habitat possibilities for the flora of Florida in the next several decades. And what we hope is that this information can be used perhaps by managers of conservation areas to help at least try to augment those plant resources that we have and prevent against the total loss of the species diversity. Now, I presented this information from the perspective of the plant diversity, but the same sort of analyses could be conducted for any other sort of group of animals, again, using specimen data from the museums. And we're currently in the process of collaborating with our colleagues in the fish range to do similar sorts of analyses on the fishes, and also with some of our colleagues who study the butterflies and moths of the state to investigate what their responses might be as well, particularly in combination with what might be happening with the plants. Now, these tools and methods are portable, not just to other groups of organisms in the state of Florida, but certainly this whole methodology is something that can be used at any level on a worldwide basis. And this map just shows the biodiversity hotspots that have been recognized. And these could be targets for where this sort of analysis could be done. Now, I'd like to point out that, however, to get to this point of having massive amounts of data mobilized, we really need collaborations, partnerships. And it's not just partnerships among scientists. Partnerships with citizen scientists are actually leading to new sources of data and new resources that can be used for these sorts of questions. So an example of that is a wonderful tool called Notes from Nature. And this is a transcription tool that allows anyone to access a set of museum specimens and provide the transcriptions that are then associated with images. So for example, there are 4 collections that have been posted on Notes from Nature, and there's a herbarium one, a bug one, birds, and also fungi. And for whichever one suits your fancy, you can specify, and then you'll be given essentially a label, and you're asked to put in the scientific name, the locality, the date, etc. And then that information then gets fed back into the website, and then that information is linked up with the image of the specimen and then is returned to the herbarium of origin or the collection of origin. And this then allows for many new specimens to actually go online as specimens with data. So there are a lot of things that can be done with specimen data. Most of these actually focus on the locality, but many of them also focus on the date. And as I mentioned before, there are a number of these different questions that can be addressed using a combination of the information that's provided just on that label. But what can we do with the specimens themselves? Certainly there must be more to the value of these specimens than simply the data involved in the label. And so what I'd like to do during the rest of my presentation is focus on how these specimens can be used in genetics and genomics, and also briefly in ecology, and then wrap up with some applications to conservation. Now, a huge effort in evolutionary biology is the reconstruction of the tree of life. And the plant branch of the tree of life is certainly one of the research foci of our lab. We're interested in how plants have diversified, what their evolutionary relationships are, and what processes have led to the vast diversification that we see. And certainly specialists in other groups of organisms are likewise interested in those same sorts of questions, but from the different phylogenetic perspective. So you can think of this as evolutionary history or phylogeny, And I like to use the family tree metaphor, where we have the House of Windsor here, where we show basically the reconstruction of the family tree. So this is essentially what we're trying to do when we reconstruct the tree of life. Now, in fact, Darwin's Origin of Species had a single figure in the book, and that single figure was this. And this is essentially a phylogeny, or evolutionary history, of a potential putative group of organisms. Now, shortly following Darwin's publication of the Origin, other scientists grasped onto this tree of life concept, and this was a rendering by Ernst Haeckel, produced in 1867, showing the vast diversity of life summarized in this tree-like figure, with the plants on the left branch, the animals on the right, and then various single-celled organisms in the central part of this tree. Now, over a century or more following this, most scientists recreated evolutionary history primarily using the morphology or general attributes of the organisms. But for the last several decades, we've been using DNA data to build these trees or reconstruct these histories. And essentially, all that's required is to have a sequence of DNA from each of several species, and we can compare these DNA sequences for similarities and differences and use those attributes then to build a phylogenetic tree, or an evolutionary rendering of this historical relationship. Now, we can go from this simple sort of tree to a much more complex one. This one includes a couple thousand species. And this, again, is just the tip of the iceberg. We are over here somewhere. This is the animal branch on that side of the tree. And I think that this is a very humbling way to think of ourselves. We are no more important than any of the other tips on this tree. We're simply one of those tips. And then plants are here. So this will be this is the clay to focus for the next several slides. And I've expanded this out a little bit to illustrate the green plants, this branch of the tree of life. As I mentioned earlier, there are about 500,000 species of green plants. They have a very deep evolutionary history. They go back about 1 billion years. But most of the plants that you're most familiar with are the flowering plants, and they fall within this clade here, which is a relative newcomer in evolutionary time, originating based on the fossil record about 130 million years ago. Now, we certainly know a lot about the general features of the evolutionary history of the plants, but there are many, many details that are missing. And in order to fill in those details, we can make great use of our natural history collections. Now, these natural history collections are not simply the physical specimens of the organisms themselves, but in fact, in many cases, natural history museums, such as the Florida Museum of Natural History and many others, also have genetic resources repositories or DNA banks that have samples of DNAs or tissues that are also available for research. And so we're not limited to the observations that we make on the specimens. We can actually go to these collections of DNA and make use of them as well as we start to try to reconstruct the evolutionary history of these large groups of organisms. Now, I'd like to present an example here based on the work of one of my graduate students, Greg Stahl, because I really like the way that he has developed an integration between herbarium specimens, DNA data, and the fossil specimens as well. So he's using fossils, he's using herbarium specimens, and he's using DNA, all combined to try to understand the history of this group of plants called the Icacinaceae. So here's a picture of Greg. And the reason that he's selected this group of plants is because it has a pan-tropical distribution. You can see its distribution in South America, Africa, parts of southern India, and then also in Southeast Asia. And understanding how these plants diversified associated with the movement of the continents in particular has been particularly interesting to Greg. The plants are woody vines. You can see here this woody vine circling this tree trunk. And they have very characteristic fruits. These fruits have very deep reticulations or networks in them, and these make them very evident in the fossil record as well. So this plate shows fossil fruits from this group of plants from about 60 million years ago. So because of the very characteristic fruit features, the fossil record is extremely valuable in this particular group of plants. You can't say that for many groups of plants, but this is one particularly useful attribute. So when you look at the fossil distributions of Icacinaceae, this group, you can see all of these little stars here. Those represent fossil habitats. So even though today the plants are distributed only in the southern hemisphere, you see a number of instances of these fossils being present across North America, Europe, Northern Africa, and also in Japan. So the fossils are really valuable for his work, as are the herbarium specimens. But the herbarium specimens are not only important as a source of general features, they're also important as a source of DNA. So these specimens actually can be used as a source of DNA for doing molecular work as well. So this shows a specimen collected in 1955 in India. And Greg was actually able, despite the sort of ratty appearance of this, to get some very good DNA from this specimen. And in fact, it's possible to get DNA out of many herbarium specimens, in some cases dating back over 100 years. This makes our herbaria and our other museum collections not just valuable as resources for the label data and the morphology of the individuals, but also as a potential huge DNA bank that is largely untapped. Now, it was for several years a potential problem to use DNA from museum collections because the DNA is often slightly degraded, slightly to maybe very degraded. And in those cases, at least using previous DNA technologies, it was very difficult to make valuable use of that information. However, our current DNA technology is all based on what are called short reads. And so basically the technology, I'm not sure, I think with this light up here, you guys can't see this too well, but the reads are very short. They're just a small number of base pairs, but we get lots and lots and lots of copies of DNA sequences. And when these things are generated, they're lined up in overlapping fashion. And so that even though you start out with short reads, you can concatenate them into long reads so that you get a very long intact DNA sequence. So what this means is that we can actually use degraded DNA because that's all that this procedure actually requires. And so our new technologies are allowing us to make tremendous use of our museum collections from a molecular perspective. So back to Greg's work a little bit. He's using this information from herbarium specimens to do genomic analyses on plant specimens in this particular group of plants. He uses the DNA sequences to reconstruct the evolutionary history, and he's just in the process right now of analyzing the data. So he's not quite at this point of having the tree put together. But I really like the way that he's pulling these different resources together. And I think this is what the future of our natural history collections will be, an integrated opportunity to use morphology, paleontology, and molecular data all coming together to address particular questions. Ultimately, allowing us to pursue in much more detail the evolutionary history of life. And through doing this, we can then understand better what the processes are that have yielded this diversification. So I'd like to just mention briefly an application to ecology. And that is that we can also use our specimen data to make inferences about the roles of species in the ecosystem. So this is particularly, again, something that plant biologists are investigating. It's possible, in fact, to take our red maple example again, to take our herbarium specimen and make some inferences based on leaf characteristics about the role that that species might be playing in a given ecosystem. And so now through image analysis, we're starting to be able to use herbarium specimens in yet new and exciting ways in ecology that we previously were not able to do. And finally, if we turn our attention to specimen data and conservation, I'd like to return to this area of Florida called the Lake Wales Ridge, located here in the central part of the state. Now, this is an area that is characterized by having many rare species. There are plants, there are birds, there are insects, there are all sorts of organisms, lizards, that are endemic to this one very narrow ridge. However, this ridge is under tremendous threat due to habitat loss. The area has been converted, first of all, to orchards, then to golf courses, then to retirement communities. And all of these sorts of land use changes have altered the native fire regime. And this is an area that is very, very dependent on fire for rejuvenating the vegetation. And so even those parts of the ridge that are still intact have suffered because of having this altered fire regime. So one of the most rare species is the species called Ziziphus salata. And our lab, and in collaboration with groups at the University of Central Florida and at Archbold Biological Station, have been working for many years to develop conservation and management practices for this species. Likewise, there are many other plant endemics, a few of which are listed here, that also have some conservation plans underway. The problem is you can't effectively conserve a large number of species through independent sorts of species-specific conservation plans. And so we've been thinking about how best to scale up these activities so that it's not one species at a time. And we think that one of the ways that we can perhaps best do this, oops, Microsoft PowerPoint has encountered a problem. Okay. We'll open that back up. I think the computer's saying we've had enough. We've only got a couple more slides. Here we go. So if we return to our analyses using the label data to make predictions about ecological niches and the future distributions of these species, we could start to think about doing this on a community level. So we could actually do these sorts of analyses for all members, plants, birds, lizards, insects, et cetera, all that occur in a given area, and then do some sort of an integrated analysis over time to hone in on perhaps the best conservation strategies that could be used. So again, we have projections for 2050 and 2080 for these plant species, but why not do this for the groups of plants, particularly in a threatened area, and also the other organisms that occur there as well. So I hope I've been able to convince you of the great value of natural history collections. Certainly, they maintain their utility for systematics and taxonomy. We wouldn't want to let Linnaeus down. We're still doing the same sorts of work in terms of specimens and species descriptions that we were doing as a community centuries ago. But of course, these specimens now have taken on many new sorts of values, ranging from their resources as genetic banks, their opportunities for telling us more about what might happen under models of climate change. They allow us to reconstruct evolutionary history and make inferences about the processes that have given rise to diversification. And they're also valuable as sources of data for conservation purposes. So with that, I'd like to thank all of you for your attention and thank my colleagues at the Florida Museum and at IDIG Bio and a few other folks who've contributed a lot to what I talked about. So thank you very much. So, and I'm happy to take any questions if anyone has any. Yes?
This is a pretty specific question, but I noticed over there on those specimens and also on the sheet that some of the dates, you have 2 different dates. So I was just curious from a collections perspective, one of them is the collection date and the other one is?
So a lot of times what happens with a specimen is that as an expert comes through the collection and uses that specimen or studies that specimen, he or she will put an annotation label on the specimen as well to say, you know, this is my name, I examined this specimen on this date, and I agree that this is the correct species name. Or you might put on it an annotation label that says, I removed a piece of this specimen for DNA analysis, or I removed a piece for biochemical analysis, or something like that. So if a specimen is quite old and has been used a lot of times, you can end up with you can actually track the history of the use of that specimen. And of course, all of that information would be great to have online as well, so that we have a record of how a particular specimen has been used. So, yes?
I was particularly interested in your map of the biodiversity hotspots. I think Greg did that work, didn't he? Your postdoc, Greg? Or?
So the biodiversity hotspots map, that's actually, I think it might be World Wildlife. I'm not sure. It's one of those organizations, which is probably in the small print on that.
I think it's fascinating, and I realize it's tied in with projected global warming, but do you recall the common thread? Because they seem to be spread out, for example, around the U.S. I particularly focused in on the Pacific Northwest. So what's the common thread that would make them?
Yeah, so let me go back and we can put that up. Let's see.
I was going to ask you to do that, but I wouldn't have asked you, so thank you.
Yeah, there we go. Yeah, whoops. There we go. Yeah, so a lot of times it's a combination of various geological traits, climatic traits, things that are basically that allow for diversification to occur. And so that's why we see things like, so yeah, there's one in the Pacific Northwest, and actually it's really the whole sort of western part of the US that is included there. There's the California floristic province is highly species-rich area in terms of plants and many other organisms, and that's related to conditions such as varied topography, varied climate, et cetera. And that yields processes that can just lead to rapid diversification. We also see the same sort of thing, like in the Caribbean, a lot of times where there's geological activity and island systems as well, those often interact to produce lots of species. So those would be just a couple of explanations, I think. Yes?
Do you work on any carnivorous plants, and if so, how many?
Oh, that's a great question. So I had a graduate student who worked on carnivorous plants, and he was particularly interested in pitcher plants. So I had a picture several times, I think you might have noticed, of this pitcher plant on the left. So that's one genus of pitcher plants, but pitcher plants have actually evolved multiple times in the whole history of the flowering plants in very unrelated groups. And so it's an amazing set of what we would call convergent evolution, where the same, kind of what looks like the same structure originated multiple times independently. And my graduate student was interested in whether the genes involved in making those pitchers were the same in these very different groups. And he never quite solved it, but he learned a lot about the genes that help to make a pitcher out of a regular leaf.
One more thing. Are carnivorous plants very common in the plant world?
Well, they're really found in kind of 4 different places. If you think of like the plants as being this one great big huge family tree, there are like 4 subfamilies in that family tree where you have carnivorous plants. And then within each one of those subfamilies, you get various types of carnivory. So sometimes you get the pitchers, sometimes you get things like the Venus flytrap, that actually closes around some insects. And then sometimes you get some other mechanisms that are either more active or less active than those. But those are kind of 2 of the most common ones. Yeah, thanks for your questions. Yes?
Are any individual natural history collections able to prioritize saving, like storing DNA for some of the older specimen before a research project comes along that will need that stuff? Because obviously this timeline of decay is ticking.
Right,
so I'm just curious, are there
So
initiatives?
unfortunately, the resources for developing these DNA banks and the natural history collections are limited. And so no one's able to have this sort of comprehensive collections in their molecular collections that they have based on their other specimens. So, so we're always behind. What we're hoping will become best practices is that as people make new collections of specimens, they're also making new collections for the DNA bank so that you end up not having a backlog into the future. Certainly some collections are going through their more important collections and trying to save snippets of, particularly for plants, it works better, I think, than for most animals. A lot of the animal tissues don't hold up as well in their preservatives as the plant tissues do. But yeah, I mean, it would be, with more resources, more collections would be doing that. Yes?
If I understood correctly, you are thinking about conserving ecosystems rather than individual species. I love the idea, but how will it be done?
Yeah, well, that's of course, a really important question. I think we're finding that saving individual species is not probably going to work unless we can save components of their ecosystems. And so the best way to save anything is going to be through saving larger areas of land so that we can preserve some of the processes that take place within those areas. I've been pleasantly surprised. So I've lived in Florida now for not quite 14 years. And I've been pleasantly surprised at the general attitude in the state toward preserving natural areas. So despite this 1,000 new people per day moving there, and this tremendous influx of people and urbanization, there's a strong regard for any areas that are still natural. And so the state government has been purchasing a lot of these over the course of the last couple of decades. And so, we're hoping that at least some of that can help in that particular case. The problem is that the patches are often very small, and whether that's enough to save anything, I think, is still an open question. So, yes.
This is more an observation. I'm impressed from today, 2050 and 2080. And I would observe they're using tools that are probably less than 30 years old. And are you hopeful that there are some new tools beyond putting things in the cloud or the various things you're doing with DNA that might foster this? Or is it hard to make a crystal ball that looks and says, oh, I only wish we could do such and such?
Right, yeah, so that's a really good question too. I think it's, I guess the perspective that I'm having on all of biodiversity is that we need to save as much as we can, not just in terms of naturally, but in terms of our specimens. And that, will allow us to maybe develop methods that we can use for other sorts of work. But in terms of what's ahead, boy, I don't know. Things have moved so fast in the last few years, it's kind of hard to imagine what might be next. Yeah.
interested in the modeling, and since you have a number of inputs into the model, and these things change over time, what are your plans for updating the models with new data that comes in? Because obviously you're extrapolating.
Mm-hmm. Yeah, so yeah, another great question. So what we've been working on recently is developing models that don't use, so that the way that these niche models have worked in the past is that they basically rely on a 50-year average of temperature for a given location. So, you know, if you have a point and you want to find out what the characteristics were of that point, you say, okay, what year was that collected? And then there's a 50-year average that, you know, kind of is associated with that point to figure out the precipitation, the temperature, et cetera. But what we've actually done is say, okay, if this plant specimen was collected in 1932, what were the characteristics like in 1932? And there are actually databases for the US that go back very far in terms of precipitation and temperature. And we can build the models based on what the characteristics were for the particular year in which the specimen was collected. And this actually allows us to make the models much tighter than using these sorts of bigger averages. So that's one way that I think that we're becoming more able to improve the accuracy of the models. Certainly, we've done these future forecasts based on several different projections of CO2 emissions, an optimistic one and a pessimistic one, and then each of those has, I think for each of the optimistic and pessimistic, there are like 3 different additional categories. And so as those models and those predictions for what the CO2 levels might be become more resolved, we'll also be able to probably update the models as we go. So basically, we've been developing methodologies that allow you to plug in your locality data and make these other specifications and then get your model results out the other end. And that will be, those will be usable regardless of what the actual other components are, so that it should make it easier to do those updates that you're suggesting. Mm-hmm.
Okay, let's wrap this up, but before we give Pam a big hand, please, there's a display, a little pop-up display over here. For those of you that have never actually seen herbarium specimens up close and in person, you can actually take a peek, all right? And there's more than you think you can glean from these flattened and dried specimens than you might believe, actually. But with, so otherwise, let's give